Good day, listeners and viewers. I am Adenuga Ajibola, and I'm here to take these biology lessons with us today. And today we are going to be looking at plant reproduction. What is reproduction in the general sense? Reproduction is a process through which all living things produce new individuals of their own kind. The process through which living things produce new individuals of their own kind is what we call reproduction. Through the process of reproduction, living things are able to perpetuate their species. That is, they are able to regenerate, produce organisms of their own kind for life to continue. So as plants are living things too, they also reproduce. So we are going to be looking at how these plants produce their own young ones. In flowering plants, there are two types of reproduction. We have the sexual reproduction and we have the asexual reproduction. Sexual reproduction in the general term is talking about a kind of reproduction which involves two gametes. We have the male gamete and the female gamete fusing together to form what we call a zygote. So in sexual reproduction in plants is a kind of reproduction which is done by seeds. Then asexual reproduction is a kind of reproduction in which does not involve two parents, involves only a single parent, so a part of the parent is used to produce offspring, young ones. So we call asexual reproduction in flowering plants vegetative propagation. So in plants, there are different types of asexual reproduction. We have the vegetative propagation, we have budding, we have spore formation, that's what we call the sporulation, then we have fragmentation and so on. We have lots of them and we are going to be taking a few of them and we look through them today. The first one we are going to be looking into is vegetative propagation. In this method, new plants are produced from the vegetative part of the plant, of the parent plant, such as part of the root, part of the stem could be used, or the leaf to produce young ones. And we have different types of vegetative propagation. We have the natural vegetative propagation and we have the artificial vegetative propagation. In natural vegetative propagation, this propagation occurs when the auxiliary bud grows into a lateral shoot and develops its own roots, also into what we call the adventitious roots. Plant structures allowing natural vegetative propagation include the bulb of the plant, the rhizomes, the stolon, and the tubers. All these are the plant structures that allow for vegetative propagation. Looking at these different plant structures that allow vegetative propagation, they include the bulb. The bulb, such as that in plants like the daffodils and the onions, they form lateral buds from the base of the mother bulb. We have a bulb and we have lateral bulbs forming from the base of the mother bulb, which produce new smaller bulbs, or what we call the bulbels, in subsequent years. So these bulbels are the ones that now grow into new individual organisms. So from one particular bulb, you can have a generation of so many other bulbs and you have more plant structures. Then the rhizomes. Rhizomes are root-like stems that grow horizontally under the ground, like that of what? The ginger. New roots and shoots form at the nodes, with the shoot growing upward to form a new plantlet, that is a smaller plant. Then the lateral buds grow out to form new rhizome. Example include the iris and the root ginger. That is why you have a particular ginger, if it's kept in a cool place, you see new structures coming out from the ginger roots, then it grows into more plants. Then the other structure which allows vegetative propagation is the stolon. The stolons, also what we call the runner, are horizontal stems that grow above the ground, for example in strawberries. We have tiny plants that form along the stolon as they're extending outward. Then the root will form where it touches the ground. So you have a particular plant, you have a stem that will grow from that particular plant and extend outward. As it extends outward on the surface of the ground, where it touches the ground, you have the formation of a root. And when that root forms in the ground, then you have what? A new plant. Then when the connection with the parent plant breaks, the new plant can now become independent. Therefore, it will be taking up nutrients, it will be carrying out all life processes on its own from the parent plant. Then the other structure that allows for vegetative propagation is the tuber. The tuber are swollen portion of an underground stem. So tubers are also stems. And this stem stores food so as plants can lay dormant over the winter. So when the plant is not getting enough nutrients, for example in the potato, it stores the food in the tuber so as to get nutrients when it's not getting enough from the soil. So from this tuber you have axillary buds, which we commonly call the eyes. It will form over the surface of the tuber. When it forms over the surface of the tuber, it will produce a new shoot. That is, another new shoot will start growing. Then it grows into a new plant in the following year. So if you have a potato at home, you keep it in a cool place, you can have tiny eyes. There you now have new shoots coming out from the main tuber. Then you have a new plant from the tuber. Then another structure, we have the combs. The combs are enlarged bulb-like structure which forms underground. They are enlarged bulb-like underground stem. These vegetative structures store nutrients in fleshy solid stem tissues and are typically externally surrounded by papery leaves. You have kind of leaf structure around this. Due to their physical appearance, combs are commonly confused with bulbs because they look so much like bulbs, but they are not bulbs. The major difference between the bulb and the comb is that the comb contains a solid tissue internally, while the bulb have only layers of leaf. Like the taro has what we call the comb, and the onion have what they call the bulb. If you look at the structure of the onion, it's just like layers of leaf over one another. So comb produce adventitious roots and possess buds that develop into a new plant shoot. When this develops, you now have an individual plant. Plants that develop from combs are the crocus, the gladiolus, and the taro. All these are plants that develop from combs. Now we are going to look at artificial vegetative propagation, because we've looked at natural vegetative propagation in that those structures can happen on their own. But when we talk about artificial vegetative propagation, these are man-made or man-affected vegetative propagation. Here you have horticulturists and gardeners. They are the ones that use what vegetative propagation method to produce plants. And this method involves taking a piece of one plant that we call the parent plant and causing it to regenerate itself into a new plant. So we could have a particular plant and I create some effect on that particular plant to bring about a new plant which is similar to the parent plant. Common methods which are used in artificial vegetative propagation include the cutting, grafting, budding, and we have tissue culture. Methods of vegetative propagation, that is the artificial, include the stem cutting, or what we refer to as cutting. We have grafting, budding, and tissue culture. Looking at stem cutting, or what we refer to as cutting. In cutting, we take a part of a plant, typically the stem or the leaf of the plant. It is cut off and replanted in the soil. When it is replanted into the soil, we have adventitious roots, new roots we develop from the cuttings, and a new plant is formed. Cutting can be treated with hormones. The cuttings can be treated with hormones before it is being planted to induce root development in order for it to germinate faster. Example of crops or plants which we use stem cutting for are sugarcane, cassava. When we look at the cassava, for us to regenerate cassava, if we want to have a cassava field, we take the stem of the cassava that have been harvested, then we cut it into bits, then we put it into the soil. When it is being watered and it gets sunlight under the right condition, you have what? New stem coming out from the old cassava stem. So you have stem and leaf, then you have regeneration. Same thing with the sugarcane. The sugarcane stem can also be cut, put into the ground, and you have the germination of a new plant. Another method which is used in vegetative propagation, that is the artificial vegetative propagation, is grafting. During grafting, a desired cutting, or what we call the scion, is attached to the stem of another plant. We call the stem of a particular plant, which we call the scion, and this is attached to the stem of another plant that has still been in the ground. It remains rooted in the ground. The tissue stem of the cutting will become grafted into or integrated into the stem system of the base plant. Over a period of time, then it starts growing. So it's just like taking a particular plant, we cut the stem and attach it and bind it to the structure of another stem which is having a root in the ground. Then this starts germinating. Then another method which is used in vegetative propagation is what we call layering. During layering, the plant is bent. It involves what? The bending of a plant branch or the stem so that they touch the ground. It's just similar to that of the runner, or what we call the stolon, that is in natural vegetative propagation. But in this case, it is the plant that is induced. We induce a plant for the stem to touch the ground. When it touches the ground, the part in contact with the ground will be covered with soil. When it is being covered with soil, over a period of time, adventitious roots will start to extend from the structure. Other than the plant roots, the plant root will develop into another plant, and this will form what? The shoot with new roots, and this is known as a layer. This type of layering also occurs naturally. That's we call, in another technique, we call it what? The air layering. Here the branches are scraped and covered with plastic to produce moisture or to reduce moisture loss. The new roots will develop where the branches are scraped and the branches are removed from the tree and replanted into the soil. This is known as layering. Going further, we look at suckering, that is suckers. Suckers are generally lateral branch with terminal buds that grow at the base of the underground stem of a certain plant. Suckers, when they grow at the base, in such plants like the banana, the plantain, the pineapple, when you have tiny suckers growing from the side, then this sucker can be uprooted or cut off from the parent plant and it will be put into the ground. Then you have another new plant. In such a way, you have the propagation of the plant, in which you have a new plant from the old one. Then the other one which we are going to be looking at is tissue culture. This is a technique which involves the culturing of plant cells. The plant cells are taken from different parts of a plant, that is the parent plant. These tissues are placed in sterilized containers. When they are placed in sterilized container and nurtured in a special medium, until the mass of cells known as what? The callus are formed. This special medium in which they put different nutrients, different substances, minerals to enable the plant to be able to grow. So the callus are then cultured in a hormone-laden medium. When these callus are cultured in the hormone-laden medium, then they eventually develop into tiny plant structures which are called the plantlets. These plantlets are now the ones that will be planted into the soil and they matured into fully grown plants. We call this tissue culture. Now we look at the other type of asexual reproduction. There we talk about budding. Budding occurs in such organisms like the yeast. In budding, you have what? A small projection from the side of the yeast cell. That small projection is called a bud. It grows and becomes a new yeast. When it grows and becomes a new yeast and then it detaches itself from the yeast, then you are now going to have what? Two different yeast organisms and the parents and the offspring coming from the same one. So in such a way you can produce more yeast organisms. Then the next one we are going to be looking at is fragmentation. What is fragmentation? Fragmentation is a process in which an organism which is fragmented grows. When it grows and enlarges to an extent in which it cannot physiologically hold itself, then part of it breaks up. So when part of this branch or the fragment breaks up, it can now develop into a new plant. For example, we have what? The Sporangium. When the Sporangium plant grows up and to a point in which it becomes physiologically dependent, in which it can no longer hold itself, part of it will break, then that part we call the fragment will now germinate and grow into an individual Sporangium. Then sporulation, or what we call spore formation, occurs in organisms like the fungus and the ferns. In this method, fungus produce a ball-like structure which we call the sporangium. The sporangium contains tiny cell-like structure which are called the spores. The spores in the sporangium will keep on growing. Then when it becomes fully grown, it bursts open. Then the spores are dispersed. When these spores get dispersed, then they can germinate into new organisms where they have been dispersed into. Therefore, you have the multiplication and the regeneration of organism of that species. So what are the advantages of sexual reproduction? Advantages of sexual reproduction include the population of the organism can increase rapidly when the conditions are favorable. Like, for instance, you have the spores. One sporangium contains millions of spores. So when it contains millions of spores and it bursts open, you have these millions of spores being dispersed. As they are dispersed from one organism, you could have so many others. So therefore, you can have a high population increase when the conditions are favorable in a very short period of time. Then another advantage is that only one parent is needed for the propagation to take place. It does not involve two parents, so only one parent is needed. Therefore, from one parent, you can have so many offspring. Then, it is more time and energy efficient as you don't need a mate for propagation. It is more time and energy efficient as you don't need a mate. So it saves time and energy. Then it is faster than sexual reproduction. Another advantage is that desirable characteristics of parents are being transmitted to the offspring. So take, for instance, if you desire a particular character from a parent and the plant is being produced asexually, those same character in which you get from that parent, you get the same character in the offspring because it's from only one organism, so there is no mixing of genes whatsoever. But as it comes with advantages, it also comes with disadvantages. The disadvantages include it does not lead to genetic variation in population. So you are going to have, just when a new plant is produced from the old one, like the one which we talked about, the dispersal of the spore. So all the new spores that will be produced, we have the same genetic combination. There will be no variation whatsoever. It's just like having just one plant a million times all over. Therefore, there will be no variation. Then the species may also be suited to one habitat because there's no mixing of genes, so they might not be able to survive in other habitat. So for propagation to take place, it might be hindered when they move to habitats which they are not well suited for, because there is no genetic variation. And because of inadequate genetic variation, the diseases may affect all the individual in the population, because they are the same plant. When one plant is affected, therefore all the other plants are subject to the disease, in which you can have all the plants on the field being wiped out in a short period of time. These are the disadvantages of asexual reproduction. Now we are going to go into sexual reproduction. Sexual reproduction is a reproduction which involves the fusion of nuclei of a male and a female gamete to form a zygote. When we talk about fusion, it means joining together. So when you have a male gamete and a female gamete joining together, they form a zygote. This process is known as sexual reproduction. The sexual reproductive organs of higher plants are known as flowers. So it is the flower of the plant that enable the plant to undergo sexual reproduction, because flowers come with various structures which include the male structures and the female structure. There we have the anther and the filament forming the male structure. Then we have the stigma, the style, and the ovary, which we call the carpel, which forms the female structure. Then we have the petals, which is the colorful part of the flower. Then we have the sepal, which is a leaf-like structure from which the petals blooms from. Then the receptacle and the ovule are also part of the flower. So there are four commonly distinct wholes of a flower part. We have the first whole, we have the outer calyx, which consists of sepals. That is the leaf-like structure. Inside there lies what we call the corolla, which contains the petals. There we refer to that as a Andriosium, or group of stamen. And in the center, we have the Ginocio, which consists of what? The pistil, which is the male reproductive structure. The sepals and the petals together form what we call the perianth, or what we call the floral envelope. The sepals are usually greenish and often resemble reduced leaves, while the petals are usually colorful and showy. So the part of the flower that is majorly colorful is the petal, and the sepal is that green structure. In most cases, you find underneath the structure of the petal. Then the male structure, which we refer to as the Andriosium, comprises of stamen, and each stamen consists of supporting filament and an anther, in which we have the production of pollen. So the pollen grains are produced in the anther. Then the Gynosium is referred to as a female reproductive structure of the flower. It comprises of one or more pistils, each of which consists of an ovary with an upright extension, which we refer to as a style. And it is on the style we have what? The stigma, which is the pollen receptive structure. That is the part that receives the pollen grain. So a flower having petals, sepals, stamen, and pistil, when it has all these four structures, we refer to it as a complete flower. So a complete flower is defined as a flower which possesses the sepal, the petal, the stamen, and the pistil. When a flower has some of these structures but not all, we refer to it as an incomplete flower. So when a flower lacks one or more of either the petal, sepal, the stamen, and the pistil, it is called an incomplete flower. Then the stamen and the pistil, when they are not present together in all flowers, because they are not present together in all flowers, when both are present, we call such a flower a perfect flower. So a perfect flower is referred to as a flower which contains the stamen and the pistil together on the flower. And we also refer to it as bisexual, that is containing the two sexual structure. Then, regardless of the lack of any other parts which renders it incomplete, so we can have a perfect flower which is incomplete and also complete. Then a flower that lacks the pistillate, a flower that lacks stamen is referred to as pistillate or the female, while the one that lacks the staminate is referred to as the male. So when a flower doesn't have pistil, in which the flower contains only the stamen, we refer to such flower as a female flower. Then when we have a flower which lacks the staminate and contains only the pistillate, that is the pistil, we refer to it as the male flower. When the same plant bears unisexual flower, that is, when a particular plant bears a flower that is only having the male structure and the other flower on the same plant as unisexual, is also unisexual. Let's say a flower is having male and another flower is having female on the same plant, we call such a plant monoecious or a hermaphrodite. Such flower has, or such plant has a flower in which some of the flowers are male and some of the flowers are female. These are called the hermaphrodite plant or the monoecious plant. Example of monoecious plants are the oil palm, which is used to produce palm oil, the oak tree, which is used in furniture, then we have the corn, or what we refer to as the maize commonly. Then another situation we could have arising is when the male and the female flowers are on the same plant. This is referred to as dioecious. This is referred to as dioecious, which we also refer to as unisex. Example, the date plant, the pawpaw, then we have the cottonwood, then we have the willow plant. All these are dioecious plants. Then when they are male, female, and bisexual flowers on the same plant, that is, there are some flowers that are male alone, then there are some flowers that are female, and there are some flowers that are having both male and female structures on the same flower. Such flowers are called the polygamous plant. Such we call polygamous. So a flower may be radially symmetrical and it could be bilaterally symmetrical. So example of radially symmetrical flowers include the rose flower, and we refer to this as regular flowers or the actinomorphic flowers. We refer to them as the regular flowers or the actinomorphic flowers. Then bilaterally symmetrical flowers like the orchid and the snapdragon are referred to as irregular flowers, or we refer to it as zygomorphic. So what are radial symmetric? When a flower is radially symmetrical, that means when the flower structure is being divided along more than one vertical plane, it can give mirror images. when it's divided along more than one vertical plane, it can give mirror images. But bilateral symmetry is such that when the flower is being divided, it can only be divided along one vertical plane to give mirror image. When it's divided along more than one, it would not give mirror images of itself, so we refer to it as bilateral symmetry. Now we look at pollination, because for reproduction to take place, we need to have what we call pollination. Pollination is defined as a transfer of pollen grain. It's a transfer of pollen grain from the anther to the stigma of a flower. From the anther to the stigma. There we have the stamen and the pistil, which are the male and the female reproductive structure of the flower. They are the ones directly involved in the production of seeds. The stamen bears a structure which we call the microsporangia, which are spore cases in which are developed numerous microspores, which are developed into numerous microspores, which we refer to as potential pollen grains. Then the pistil are the ones that bears the ovules, which encloses the egg cells. When a microspore germinates, it is known as the pollen grain. Then when the pollen sac of the stamens anther are now ripe, the anther releases them and the pollen is shed. I repeat, when the pollen sac in a stamen's anther is ripe, it will release the pollen and the pollen will be shed. Then fertilization can occur only if the pollen grains are transferred from the anther to the stigma of the pistil, which we know as pollination. So in general term, pollination is the transfer of pollen grain from the anther to the stigma. And we have different types of pollination. We have two major types of pollination. We have the self-pollination and we have the cross-pollination. Depending on the structure of the flower, we could have cross-pollination or self-pollination. Self-pollination is defined as a type of pollination in which the stigma. Self-pollination is defined or is a type of pollination of a stigma by the pollen from the same flower or another flower of the same plant. When the stigma is pollinated by pollen grains of the same plant or the same flower, we refer to it as self-pollination. Self-pollination occurs in many species, but in the others, perhaps the majority. It is prevented by such adaptation as the structures of the flower, self-compatibility, and the maturation of stamen and the pistil of the same plant or flower. They develop at different times. Here we look at what are the limitations or what are the things that could prevent self-pollination. Like some flowers, the stamen and the pistil do not develop at the same time. Therefore, it will be difficult for the flower to pollinate. That is why some flowers don't self-pollinate. So, looking at the other type of pollination, we say cross-pollination. This is a transfer of pollen from the anther of a plant. This is a transfer of pollen from the anther of a flower of one plant to the stigma of the flower of another plant. So that's just a transfer of pollen grain from the anther to the stigma of one plant to the other. Self-pollination is brought about by different agents. We could have insect-pollinated flowers or wind-pollinated flowers. So when we have wind-pollinated flower, what are the things that make the plants to be able to undergo wind pollination, or what are the limitations? The limitations include the lack of colors, odor, or nectar. These are the same factors that enables the plant or the other plants, such as the animals, to pollinate them, like the structure of the plant, of the flower, the colors, and they produce scent. So we have the insect-pollinated flowers. Insect-pollinated flowers are conspicuous flowers by virtue of their structures, the color, and the production of scent and nectar. These are the things which attracts the insects or the animals to them and enables the insect or the animals to help transfer the pollen grain from the anther to the stigma. So on this note, I will leave the topic at this, and in our next class we continue from this part. Please remain safe and stay blessed. Thank you.